Hi, Year 12s, welcome back. So we're going to go through our next PowerPoint today. We're going to do a quick recap of PCR. All right, remembering PCR is just an amplification technique for DNA. All right, all we need to do is get our small amount of DNA into many, many different strands of DNA, and then we can use that after that. All right, there's this three main steps. We have denaturation, okay, which is the DNA splitting. All right, and that's when it's brought up to a temperature of 90 to 95 degrees. All right, we have the second step, annealing, which is where the DNA primer will attach to the single-stranded parts of the DNA. Okay, and that happens around 50 to 55 degrees Celsius, so a lot cooler. And lastly, we have the elongation or extension stage, where the Taq polymerase binds to the DNA primer and then will extend the strand to create two different DNA strands. Alrighty, so in today's video, we're going to continue on with DNA separation, but we're going to look at gel electrophoresis. So these are these gel runs that you see in the image here. Again, our dot points as per usual. We're moving along, which is good. So our gel runs here are used to essentially just measure the size of DNA fragments. Okay, it's typically used after the PCR has occurred, so polymerase chain reaction. So you'll have lots and lots of DNA that you can put in each of these different wells, okay? Or many different people's DNA. It depends what you're looking at. The gel that's a part of our gel run is made of agarose jelly. So it's a sponge-like jelly that's filled with these tiny little pores that allow for movement of the DNA. Okay, so the DNA essentially gets pushed through it. All right, the agarose gel is immersed in a buffer solution to keep the pH regulated, as well as there's an electrical current going through it. So step one of gel electrophoresis is firstly you place the DNA samples in each of the wells. Okay, so DNA sample 1 or A in this example, person B, all right, and person C in the purple. All right, so you can see their DNA's in there. All right, so we also have a standard ladder which is at the side, and that's used as like a measurement device to measure a similar amount of base pairings as the DNA strands. Okay, so for example, this pink one here is roughly 15 base pairs. Okay, just it's a random number there, but that's what we use as a measurement tool. Step 2 is adding your electrical current. Okay, so we have one positive and one negative. Okay, and they're done through electrodes. So the negative electrodes go through from the well side. Okay, so these are your wells again, where the DNA will be placed. We have our ladder on the side here, and then we have our negative electrode. Okay, and essentially what that does is it pushes the DNA across. So slowly the DNA will move through the agarose jelly, through the pores, and that'll go towards the positive electrode. Okay, why is that the case? Essentially DNA is negatively charged because of one of the oxygens having an extra electron. Okay. So the important thing to note, so step 3 here is that as the electrical current goes through, okay, it's going to push the DNA fragments, as I mentioned in the last slide, pushes them across. All right, but why is one of the DNA fragments here and why is one here? Because of course, if it's a smaller DNA fragment, it's going to move faster through the gel, and it's going to be the same period of time, obviously, until we turn the electricity off, and therefore it essentially is going to travel further away from the negative electrode. Okay, whereas the larger pieces of DNA, so a longer strand, for example, with more base pairs, it's not going to go as far. It can't get through those pores as easily. It struggles to, be pushed through as something would, and therefore it's going to be closer to the negative electrode. Okay, and therefore after that we can compare these bands to one of the standard ladder we have at the side, and we can see how many base pairs and the size of the DNA strand. So step 4 is one of which we want to identify the DNA. Okay, so the DNA is quite difficult to see with the naked eye. So what they do is they added a fluorescent dye to each of the wells. Okay, and from there the DNA will then change colors. And that can be any substance, but in this example, it's ethidium bromide, which allows it to go this fluorescent pink color. Okay. From there, they put it under UV light, which is the purple you can see in the image. And then after that, we can use the standard ladder on the side to compare where these bits are to where the DNA has been found on the gel run. Okay, and from there we can make an assessment as to whether that is the same DNA from the same person. Okay, where this would be important would be, in a forensics environment, when you've got a crime scene and you found either a drop of blood or, a hair, and you've extracted the DNA from that. Can you prove that this person was at the crime scene? So if this was person A, maybe B, C, who can you prove that they were actually at the crime scene? Potentially person A in this example. All right, that's where it's useful. So just to cap it all off, to have an overall summary, we'll go through the 4 steps once again. So the first step here, as you can see, is a micro pipette, which is obviously the researcher putting in the DNA samples into the wells. Okay, we have our agarose jelly waiting to go and our 2 electrodes. Okay, second step, you add the electrical current to the electrodes. So negative to where the wells are, positive to the opposite end of that. Okay, so we're pushing our DNA away from the negative and towards the positive. The third step here, all right, you're able to finally see that the DNA is moved. So then you turn the electricity off, okay, and the DNA has gone different lengths. All right, obviously the largest pieces of DNA are closest to the wells, and the smallest DNA fragments are further away from the wells. Now, last step is you put that under UV light with different dyes. So example, ethidium bromide, which will make it that fluorescent pink, so you can really clearly see it. And that's it. That's the whole process of gel electrophoresis. It's a beautiful thing, isn't it?
Yep, it is.
All right. See you later, everyone. Have a good one.